did the introduction to your sarcomere, the muscle, and that's where we're going to start. You need to talk to me about the take-home test on the during the break. I'll give you your test. You have a test on Thursday, but instead of taking it here, I'm sending it with you today. So you have to bring it to me on Thursday. I think we talked about this one, right? The actin and myosin. So remember, where is that? Okay, so this one, we talked about what are the different, what's the ingredients that make up a typical muscle cell. So we saw the nucleus, the mitochondria, all of this. We studied the different organelles inside muscle cell. And I'm sure you remember when you looked at your muscle slides, it looked like this, right? You know that the bulk of every muscle cell is made up of these, the thick and the thin filaments. You understand? This is what's referred to as the contractile proteins, the actin and myosin. They accomplish the contraction, so you call them contractile proteins, okay? And so this one is just showing you the whatever you can see in muscle, but in reality, it looks like this. Muscle is packed with these muscle proteins, the actin and myosin, and you just see squeezed inside, interspersed. You see the mitochondria, the sarcoplasmic reticulum. You see them there. Okay, so I want to investigate further this one, the thick and the thin filaments. Why? Because they are the ones that accomplish the main action of muscle, which is to contract. They're the ones that will do that. Okay? So let's look at our thick and thin filaments. And they are really, see, they're arranged like this. See your thick filament? This is myosin. And there's your thin filament. I use the example of like Samson and Delilah, or a big heavyweight. Some heavyweights, they can lift light people and then just fling it, whatever. So just imagine your myosin is the thick filament, and look at how it looks like. It's referred to as resembling an inverted golf club. You see that? Okay? And you see the myosin? It's like this. See? It's always in a ready position like this. It's ready to grab what? The thin filament. One more time. This is what? Thick filament, your myosin, right? And this is what? Your actin, your thin filament. And the thick filament, it stays put. Don't forget, it doesn't go anywhere. It stays put. And it's like this, right? It's ready to grab the thin. It's so thin, it's ready to pull it towards the center, right? And so that's what happens, by the way. When it grabs the thin filaments and pulls it towards the center, then, can you imagine from end to end? Can you imagine that? The whole muscle will contract, right? But one is origin, the other one is insertion. It's going to pull, it's going to cause movement. Do you follow? Are you all following? Okay? So, normally, when muscle is resting, you follow? The binding sites of actin to myosin is covered. So there is something that's covering actin. It's covering actin from the view of myosin. You understand? So, yes, myosin is ready, but it can't latch on to the actin because there's something that's covering actin. Okay? There are two proteins that cover actin. You call it troponin, tropomyosin. Here. See? This is another picture. Look at this. And there's your actin. See? Normally, what did I say? Normally, myosin cannot latch onto actin because there's something covering it. Those are two proteins that cover it. And those two proteins are called troponin, tropomyosin. You understand, everybody? Okay? And so they must be pulled out of the way. So it's just like this one where the binding sites, let's say, is this green one, but what the myosin is seeing is this white part, which is troponin, tropomyosin. It's covering the binding site. So something must pull this out of the way to show then the binding site of actin to myosin. What pulls it out of the way like that is calcium. You see? Okay? So in the presence of calcium, this troponin, tropomyosin will be pulled out of the way. It will rotate it so that myosin will see the binding site of actin. You see? Okay? What releases the calcium? Nerve stimulation. You understand? Everybody? So that's why we go back to this picture over here. This one. And I am telling you right now that it will be to your benefit to know this slide by heart. Slide number 39. It will be to your benefit to know this slide by heart. Let's review. Look at the first thing that happens, the excitation contraction coupling, right? So the nerve will stimulate the muscle, right? By the way, our next chapter, or the next body system we're going to study is the nervous system. So you should know now that nerves stimulate other cells not by touching, but by releasing what you call neurotransmitters, like a transmitter radio. That's how they stimulate. So the nerve stimulates this muscle by releasing what you call acetylcholine. You need to remember that acetylcholine. That is the only neurotransmitter for muscles. Okay? So look at the nerve will stimulate this, that's number one. Number two, the result is a muscle action potential, meaning that stimulated the muscle. Okay? We're going to jump here. Look at the result of muscle action potential is the secretion or the release of calcium from the sarcoplasmic reticulum. And so the calcium is the key. See, look at it binds to troponin. So the troponin tropomyosin complex, it binds there and it moves it out of the way so that myosin now can see actin and it will just grab it on each end like this one, right? This must be actin. It's ready to latch and then it's just going to pull it towards the center. Muscle can contract. You understand this part, right? Everybody? Okay, very easy. Again, it will really, really be to your benefit, especially when you take the muscle test, if you know this by heart. I'm telling you. Okay? So let's look at this sarcomere in more detail. Okay? Let's look at the sarcomere.
What does sarcomere mean?
Sarcomere,
Like just the
sarco, muscle,
so like when you get a sarcoma or
in the muscle.
it's
Okay, so here is a typical sarcomere. Okay, there's your thick filament and there's your thin filament. We now studied what happens, right? How that myosin is able to latch on the actin. We studied that it requires nerve stimulation, which then triggers release of calcium, which then will bind to troponin, tropomyosin, I always use the example of a boyfriend and a girlfriend, right? The boyfriend wants to kiss the girlfriend, but then the parents are in the way, so you can't kiss the girl. The parents have to go to the next bedroom. So you call on your grandma. The grandma comes to pull the parents out. Now you can kiss your girlfriend, you see? Right? It's the same thing here. The troponin tropomyosin is like the parents. You call on your grandma, that's calcium. The calcium will pull them out. Now they're free to kiss one another. Okay? So this arrangement, you know that it's a regular pattern. It's telling me then that what happens to a one unit like this, if I look at what happens to one unit, that will tell me what happens to the entire muscle, right? Because it's just repeating units of sarcomere. So that's why the sarcomere is the smallest functional unit of muscle. Sarcomere is the smallest functional unit of muscle. Okay? So let's look at this sarcomere. Look at the dark part that we see under the microscope. The dark part is because of the myosin. Does it make sense? Because the myosin is thick. So when I look at it under the microscope, it's dark. Yes? Everybody? Okay? So that's dark. The dark part is referred to as the A band. Everybody, you understand? Okay? And of course, look at the actin, because it's thin, then it will look light under the microscope. So the lightness, sounds like I, and that's the I band. If this was going to come out in the test, I'm going to already leave the labeling for A band, I band. I will leave that for you. You just figure out what will happen. Okay, you tell me what will happen. So let's go back to this dark area. Within the dark area, there is that part there where the actin and myosin overlap. That's called the zone of overlap. Okay, everybody, you got that so far? And then there is also an area within the A band that is just only myosin. I call that the H zone. You don't need to write this down. You have it in your book, right? You just need to figure out what's going to happen, and it's very easy. Then this is the I band. And look at, there is a line here called the Z line. One Z to the next Z makes up a sarcomere. So what did we say? The way muscle shortens is because the myosin pulls the actin towards the center. So this is sarcomere at rest, and this is sarcomere that has contracted. Look at what happens. I will always ask, predict what will happen to the following areas when the sarcomere, or when the muscle shortens or contracts. I don't even use the word shorten, I'll use the word contract. Okay? And if this was going to come out, I will not give you this and this. It will just be this one, the sarcomere at rest. So you need to tell me what happens. So let's figure out. When muscle contracts, what happened to the sarcomere? Increase, decrease, or no change? Very good. Decrease, right? Look at. And then this one. It's pulled towards the center, right? What happened to your A band? Increase, decrease, no change.
Decrease.
No change. It's the A, correct. The A is the dark. And the dark is because of the myosin. The myosin remains, right? It's just, you're just pulling it. It doesn't change the dark band, the A band. What will change is the I band. What do you think? Increase, decrease, or no change for the I?
Increase.
What?
Increase.
No. Decrease, right? Look at, when you pull this, look at the I band. The light area is shortened. The I band, this one.
How come?
What is I? I is
Light.
light made because of actin, right? And so if it's full, what happens to the eye? Decrease.
Because the myosin is dark and actin is light.
Yes. And the myosin pulls the actin, so then the light becomes shorter. Okay? What happens to the H zone? Increase, decrease, no change. Huh? Decrease, right? The H zone is the part of the sarcomere that is only myosin. When the muscle contracts, this one becomes shorter because the overlap will increase. You understand? How about the length of the individual filaments, the thick or the thin filaments? Very good, stays the same. That's very important for us to remember. In the past, they thought that the individual thick and thin filaments, they thought that they shortened somehow or they folded. They couldn't figure out how does muscle shorten when it contracts. Until they looked at it and they investigated, they realized that there is no folding or shortening of the individual filaments. The actual contraction is referred to as sliding filament mechanism, where the actin slides over the myosin. Don't forget, which one slides over? The actin slides over the myosin. Okay?
What are the green things?
I'm glad that you asked that question.
Is that the coloring?
No, what is green? It's squiggly. What is squiggly?
Nerve
That's very good. Elastic fiber. It's elastic fiber. Do you remember I told you to thank you.
I have a family emergency, so I wrote a huge drop that off.
Oh, okay. Well, your test is a take home test on Thursday.